So as I've just indicated, for those who have come in now, we are talking about PCR or polymerase chain reaction, and we shall be looking at how to apply it in the diagnosis of viral infections.
We live in a moment where genetics is helping us to understand more and more of the world around us, from untangling evolutionary histories to more precisely diagnosing and treating disease. But to do all of this, we need to be able to find and examine specific pieces of DNA. One of the fundamental tools that we use to do this is called PCR, or Polymerase Chain Reaction. I'm Dr. Alex Danis with Mini PCR Bio, and today we're going to explore this important laboratory technique. PCR begins with a DNA sample, and DNA samples can be complex. Imagine the human genome with 3.2 billion base pairs. Trying to identify just one genetic sequence, which may be only a few hundred bases long, out of the entire genome of billions of bases, can be like looking for a needle in a haystack. PCR is a tool that finds that needle and then makes copies of it over and over until you are left with a pile of needles so large it's hard to find the original haystack. So how do we find our genetic needles in our haystack of DNA? A lot of it depends on the fact that our DNA is composed of two complementary strands that form a double helix. An A on one strand will always bind to a T on the other, and a C on one strand will always bind to a G on the other. This means that if you know the sequence of one strand, you always know how to replicate the other half. PCR is composed of three main steps: denaturation, which separates the two complementary strands of DNA, annealing, which specifies the region of DNA to be copied, and extension, which copies the DNA. These three steps will then be repeated over and over again. Now let's take a deeper look into how all of this works. The first step, called denaturation, involves separating the two strands of a starting DNA sample apart. This starting DNA sample is referred to as our template DNA. To do this, we heat the DNA up to about 95 degrees Celsius, almost reaching the boiling point of water. In just a few seconds, the high temperature causes hydrogen bonds between the two strands of DNA to break. After an initial longer denaturation to start off the PCR, each cycle requires a shorter denaturation, which can be as little as 10 seconds, but is often between 30 seconds and 1 minute. In the second step of PCR, we specify the region of DNA to be amplified. We do this using short pieces of DNA called primers. Each reaction will have two types of primers, forward and reverse. The forward primer will match a sequence on one strand of DNA at the beginning of the region we want to amplify. The reverse primer will match the end of the region on the other strand. Primers are typically around 20 bases long. When we cool our reaction down, the primers will anneal or stick to their complementary regions on the template DNA. Lowering the temperature allows hydrogen bonds to form between the primers and the template at areas where the sequence is complementary. We add lots of extra primers to increase the chances that they stick to the DNA rather than the DNA strands annealing back to each other. Annealing temperatures are typically between 50 to 65 degrees Celsius, but the exact temperature you use in your reaction depends on the specific length and sequence of the primers themselves. Our reaction may be allowed to anneal for as short as 5 seconds or as long as 30 seconds. Next, we heat the reaction back up again to extend or copy these new double-stranded areas where primer and template DNA have annealed. For this, we use an enzyme called a DNA polymerase, the P of PCR. The polymerase will find the ends of the short double-stranded regions of DNA where the primers have bound. It then moves along the DNA base by base in the 5' to 3' direction and adds the correct complementary DNA nucleotides as it goes. If it finds a C on the template strand, it adds a G. If it finds an A, it adds a T, and so on, creating a complementary strand of DNA. Our extension step can vary in time. When our target is long, we can extend this step to a few minutes long to give the polymerase time to copy the entire region. If we are copying very short regions, it may only need a few seconds. Extension typically happens around 72 degrees Celsius, the optimal working temperature of the polymerase. Of course, 72 degrees Celsius is hot. Our own body temperature is around 37 degrees Celsius, so if we used a typical human or bacterial polymerase, it would likely denature and stop working at these high temperatures. So instead, we use a special polymerase called Taq polymerase. This enzyme was originally found in thermophilic bacteria living in the hot springs of Yellowstone National Park, and remains stable in our reaction even through multiple rounds of heating and cooling. All of these steps happen in a machine called a thermal cycler. Here, we're using a mini PCR machine from Mini PCR Bio. We place our DNA along with all the ingredients needed for the reaction into tubes and then place them inside. The thermal cycler will heat it up and cool it down just as we tell it to. After one cycle of these 3 steps, denature, anneal, and extend, we have turned our original one piece of target DNA into 2. But we don't want to stop there. We repeat this cycle over and over again, now turning our 2 into 4, then 4 into 8, 8 into 16, and so on. The process creates new identical copies of DNA. We are exponentially amplifying our DNA. This chain of copying explains why it's called polymerase chain reaction. Most PCRs will run for about 30 cycles, which should give us about 1 billion copies of our original target sequence for every 1 copy we started with. For all of this copying to work, we need the exact right ingredients. So let's take a closer look at exactly what we put into each tube. First, we need our DNA template. Remember that this template is often a part of a complex DNA sample. Depending on the experiment, this DNA could come from almost anywhere, including a human blood sample, a water sample, a food sample, or even a bacterial swab. We also need our DNA primers, which specify which area of DNA we want to amplify. The DNA sample and the primers are unique to each experiment, but the rest of the reagents are the same in almost every PCR. We need the Taq polymerase, which will do the copying. We also need lots of spare nucleotides, or dNTPs, the building blocks of DNA. These are the molecules the polymerase will add to the growing DNA strand. Finally, all this happens in a special buffer which contains salts and ions that help keep the polymerase functioning. What we do with our new DNA copies after our PCR finishes depends on our experiment. We may insert it into a plasmid so that a bacteria will produce a certain protein that we are interested in. We may use it to do DNA sequencing so we can learn the exact sequence of nucleotides in our DNA sample. Often, we will run it on an electrophoresis gel so that we can visualize the size of our DNA fragment or confirm that the PCR worked as we expected. This ability to make billions of copies of a specific DNA sequence has made PCR a staple of the molecular biology lab for decades and allows everyone from students to scientists to investigate DNA in the world around them. Mini PCR thermal cyclers continue to make PCR accessible to users around the globe, from classrooms to remote field outposts and even up in space. To learn more about mini PCR thermal cyclers and learning labs that you can use to implement PCR and molecular biology techniques in your classroom, you can visit minipcr.com.
Okay. I hope you've picked some key points from that video. We'll now look at the presentation for today, which will break it down so that we understand a little better what happens in the PCR assay. Okay, so this will be short and sweet. It's a short lecture, will be done by before 4. I know it's a Friday, so many of you are eager to wear your weekend caps for a while. The outline for today's class, we'll be looking at how this test works, which has actually been described fairly well by that video. We'll look at the steps of PCR and the reagents required, and we'll also look at some of the types of PCR and how we apply them. I would like to re-emphasize here that a PCR allows us to target a specific segment or section of a DNA and make copies of it. And that number, making copies of it allows us to visualize. So the visualization can be what we call endpoint, which is on a gel. I'll show you what a gel looks like. It was actually in the video, but we'll look at it again. And you can also do it in real time. Rather than waiting for the end, you can also do it in real time and see fluorescence, color, or amplitude coming up, depending on how much DNA has been copied. This can be seen in real time after every cycle. So after one cycle, you visualize. There's a system for that. After the second cycle, you visualize, and that is called real time. So the technique makes copies of the same target so many times that you're able to visualize it in different ways, not with your eyes, but sometimes it may be possible if you use color to visualize with your eyes, but generally it's not by the eye, it's by the two techniques that I will describe. It has numerous applications, not just in medicine, but in many other applications. Currently, you guys may be familiar with forensics. For example, if you want to prove that somebody's DNA is on a murder weapon or at a murder site, PCR is a test that has been done for ages. Paternity testing as well, looking at different alleles or different genes in the body and comparing offspring and their father or whatever the case may be, and also for industrial use. So it's a test that has a lot of application. The gold standard for testing most viral conditions is actually PCR, because if you find the DNA of something, it means you've specifically identified that pathogen. It's very accurate because it's that accurate, and it's very specific to that specific organism most of the time. There are, of course, questions which, as a clinician, you have to ask. For example, a study, I don't know, 5 years ago, looked at barber shops clippers and found hepatitis B on them, hepatitis B DNA. And so even if this hepatitis B was infectious, we don't know because nobody did a culture. So what do you make of that test? When you find, like, for example, you test a surface, you swab your table and find HPV DNA or find COVID-19 DNA or maybe RNA, although usually we'll tell you why it's DNA for PCR. What does that mean? So that interpretation needs to come from you now, because the virus on the clippers may not be infectious. It has already been fixed with alcohol, and alcohol does not destroy DNA, it will just fix it. So when you do your viral, PCR will detect it, but if somebody is, say, injured by that clipper, that virus may not cause disease. So how do you interpret that? And why do you even do that test in the first place? If you find, for example, a swab from your table is positive for COVID-19, does that mean that COVID-19 virus is infectious, or is it just the genetic material that is there? So that is one of the challenges you need to keep in mind, that finding the pathogen there or the D genome of the pathogen there needs to be interpreted with caution. The video we just watched is the second one here. I encourage you to take time and watch it again. So what do we need to test to undertake the PCR test? The DNA polymerase. And you saw it was a specific polymerase because of the temperatures involved. And the temperatures allow us to control what can happen at what time. There are some enzymes that will be inactivated at certain temperatures. There are some that will be activated at other temperatures. So that's why we choose a specific, it's called a TAC polymerase, which is from a bacteria that survives in hot geysers or hot springs. And therefore the enzymes therein can work in our PCR. If we used human polymerases, they will be denatured and they will not function. The other thing we need to keep or pay attention to are the primers. And primers are what determines what you will test for. If you are testing for malaria, you can keep all this, but make sure the primer is targeting malaria. In a slide or two, I'll show you what that means. If you're testing for dengue, all this may be identical except the primer. The primer will have to be specific for dengue, and so on and so forth. So it's the primer that decides or determines what you're testing for. If you want to test for HIV, you have to design a primer that can detect HIV specifically enough not to miss any case of HIV, but also not to include other viruses as positive. And so they are very important, how you choose your primers. And also they have to be designed in such a way that they'll fit within the PCR temperatures. If you take a primer that is too long or has other issues, it may not attach very well to the DNA. So a lot goes into this design of primers. The nucleotides are building blocks for DNA. In essence, this polymerase will require DNTPs or deoxynucleotides. Deoxynucleotides are basically nucleotides that are in all accounts similar to the ones we have in the body. And they are used to make DNA. That's why they're called DNTPs. But I think during sequencing, I'll show you some of the differences so that you understand how modifying these bases can help us with our tests. The session on sequencing. The template DNA is from your sample. And remember, we want DNA, not the sample. There are a few tests that you can just pick the sample and throw it into the PCR and it works. But really, what you're targeting is the DNA. And many times that means you start with a sample and then you have to find ways of making that sample DNA. One thing is to extract from the sample the nucleic acids. And then if it is an RNA virus, to convert that RNA into DNA, because PCR will take the DNA template. So if you're starting with RNA, it may not work at all, or it may not work very well. And then the buffers are just to make sure the environment is suitable for this kind of reactions. I see 3 comments. Where do we get primers from now? As for you, most of the time, you will just order. You go online, look at what primers, if you're running a lab, that is, what primers are required for HIV, and you order them. But if it's a lab, they already have the primers already ordered, then they can just run the test. But you can also design. Like for me, when I'm doing my research, I look at what is available, then I can decide to design my own, or I can pick what is available and then play around with the steps. We call it optimization, so that by the time you look at the characteristics or the properties of the primers, you know what temperature to use and for how long. Do you do it for 1 minute? Do you do it for 30 seconds? And so on and so forth. So that is involved in selecting primers. The next question here is somebody talking about the sequence of the viruses. Yes, for you to choose a primer, you need to know the sequence of the pathogen. So that is, but you also have some primers that are random that can sequence anything. Okay, so is PCR done on viruses and RNA genome? Yes, it is, but you have to convert the RNA first to DNA. This means that you're getting the gist of this slide. Okay. There are often pretty other cool ways of making nucleotide sequences from scratch. Yes, these are called sequencing now, and we will talk about that in the next session. But yes, there's actually a video attached to this slide when I give you, you can take a look at. But you can do a sequence, you can generate sequences from pathogens, but you still need primers, even if the primers are not specific. Okay, the next thing I would like to illustrate is how these primers work, because really primers are what determine how good your PCR is most of the time in terms of detecting pathogens. And this here is a target genome coming from, let's say, from 3' on my left to 5'. So the primer will always attach from 5' to 3'. And if you notice, it's identical, that means it's complementary. So a C to a G and A to a T in that order. And because this is running from 3' to 5', the primer is now running from 5' to 3', because that's the direction that the polymerase will move. So once the primer attaches, we call this annealing, and it happens at a certain temperature. If you heat this, then it will separate, it will denature. Even the primer will denature. But if you cool it down to a certain temperature that the primer is able to anneal or attach, then they come together and form the double double-stranded DNA. After this happens, the RNA polymerase that is in the same mixture will attach and extend this strand, extending it from 5' to 3' in the 5' to 3' direction. And so if you started with this one copy, you will end up with 2 copies. And so for you to go to the next cycle, this is 1 cycle complete. In the next cycle, you hit this and it anneals or separates, and now you'll have 2 templates. And you can copy both templates into other copies of DNA. And actually, when you're starting, you will have at least 1 double-stranded DNA. We call it cDNA or just DNA. So because it's double-stranded, you'll actually be starting with 1 copy of double-stranded DNA, but each of these will pick either the forward primer, like this is actually the reverse. If you consider this as the primary strand, then another primer will attach from this side going this way, from 5' side to 3' direction. So the idea here is you have a primer that attaches to the complementary part of a target DNA. And that allows the polymerase to extend the genome, the strand, so that it can complete making the copy. This is explained now as a sequence of events in this image. We have the 3 major steps. One is denaturation. And denaturation is what you can see here. You start with a double-stranded DNA, you heat it, and then it comes as, it separates, and now you have 2. And then each of these will act as a template. You have the primer, maybe the forward primer attaching on this side, and the reverse primer attaching on this side. And once this attaches, the polymerase will extend by copying the strand. And each of the strand will now make a copy, and you end up with 2. And this really is the essence of PCR. This is repeated over and over and over, 25 to 35 times, actually up to 40 times. And whereas you start with only 1 copy of your template, you'll end up with billions, depending on how many cycles you do. I hope that is clear. I'll just check the comments if somebody's floating. Is RNA to DNA the job? Yes, exactly. RNA to DNA is the job of reverse transcriptase. So you are right. I have a slide of this, so you're just preempting it, which is okay. How do you know which end? You don't have to, as a person who is looking at this. You just the primers will know, because the primers are sequence in a sequence that is in 5' to 3'. So they'll attach the complementary sequence from the DNA. And because the DNA exists in both forms, it will attach in either side. So really, if you look at the DNA on this chart, it is the same DNA, it's just complementary. Both these 2 strands one will be moving from 5' to 3', the other one will be moving 3' to 5'. So if the forward primer can attach from this side, the reverse primer can attach from this side. Remember, these 2 sequences are not the same. The sequence here and the sequence here is not the same. So there'll be different primers, but one is called the forward primer, the other one is called the reverse primer. They are not complementary, they're just targeting the same genome. The primers, that is. Technically, and the copies made slightly shorter than the original? Yes. So when you're doing, when you're targeting like this short segment here, it will be part of a longer, say like the human genome, you're only targeting the gene that codes for insulin, for example, or even a smaller part of the gene that codes for insulin. So you have the entire genome, but you only take a small part. In fact, you take a part that is usually between 200 base pairs and 1,000, at most 1,500 or 2,000 base pairs, which is very short, given that a virus, on average, the smallest virus that you're going to deal with is about 4 kilobase, 4,000 base pairs, and most of the viruses will be around 9 to 20 thousand base pairs. So you're only targeting a small portion. In the PCRs I've done, most of the portions were 200 base pairs, some even shorter, to 1,000. Because when it goes beyond 1,000, the conditions of the PCR become a bit challenging because it is overworking the polymerase and it needs more time to copy the entire thing. Although now we have polymerases that work faster and they can do this within a shorter time. So you don't target the entire genome, you target a small portion of it. Now the challenge is, how do you select which portion? You have to make sure the portion you're selecting to target is unique to that organism, so that if you target a portion, it is able to tell you this is dengue. If you target a portion that is for flaviviruses in general, it can tell you this is flaviviruses. If you're targeting a portion for humans, it can tell you these are humans and not pick plants or pick something else. So do you hit the temperature only at the Firat? I don't know what that means. And maybe they're asking only at the first cycle. No, you keep hitting. There are cycles. I'll probably take that again so that we understand. And they get shorter after each round, not necessarily. It's just the first few steps where we have longer ones, but after some time they get shorter. But now I want to focus on the more important steps. How do you obtain DNA to be used from the sample? Please repeat the annealing and extension step. Now you hit after every round. Okay. Yeah, I think if your question has not been answered, we can still take it. So again, because this is an important part of PCR, I can go back and show you. You are starting with, say, one strand like this. Usually you have a DNA double strand, but we are only focusing on this one strand. And let's say this strand is running from 3' to 5'. It can also be running the other way around. And so because this is 3', it means the complementary strand will be running from 5' to 3'. It will be the opposite. This starting with 3', the other one will start with 5'. So the primer will attach on this side, and the primer is moving from 5' to 3', and after attaching to the complementary sequence, if this complementary sequence was somewhere in the middle, it would attach on that one in the middle. And then once it attaches, that primes the polymerase to extend that segment or strand all the way to the end. It will even go beyond here. If this thing is long, there's nothing to stop it unless there's a stop codon. So it will go all the way. So if that is annealing and extension, how do we apply it in PCR? These are the two copies coming from this process. So you start with a target template, which is then denatured at heating, after heating, and this happens every cycle, and then it's repeated these times. So after heating, it is denatured, which is really separating the two strands, and you end up with these two strands acting as a template. The template is primed by these primers attaching to the strand. That means if your sample did not have the genome, the DNA, the primers will not attach to anything. But if the sample is present, the DNA is present, then they'll attach. And after attaching, the extension will occur, and the extension is done by polymerase. And so what happens that you end up with, you start with 2 copies, and the 2 copies will now become 4. They've been copied, and they're now 4. That is one cycle. For the next cycle, you do the same. But now you're doing for individual strands, but remember this is happening for all the templates. So actually it's like having 4 reactions now occurring similar to this one, because you have 4 templates. And then for the next step, you have additional templates. So initially you have, let's say, this double strand. In the first cycle, you have 2 copies of double strands. Then in the third cycle, you have 8. You can see 4 copies of the double strand, which is 8. And then all the way you repeat, you keep repeating this act as template for the next one, this act as template for the next one. You keep repeating until after 36 cycles, you have, after 35 cycles, you have billions of copies, which is what you want, because you want as much as possible to be able to visualize. And the conditions for each are repeated. The denaturation, which is separating the 2 strands, annealing, which is attaching the primers, and extension, which is the polymerase copying the strands. This is done many times, over and over. until 35, 40 cycles to make copies. How do you select your primers? As I say, you decide depending on the target. This will be done in the lab for you. When you ask for PCR test for HIV, you don't have to tell them which primers to use. The lab knows, but in your mind, you should know that somebody selected the primers, and so that you appreciate the process that can actually go wrong sometimes, especially if you're running a new lab or you're troubleshooting, or even you have to open your own lab. So in this case, let's say we want to target dengue virus. We have options. We target the family. You can target a portion of the virus that is general to the family flaviviruses, but does not occur in other viruses. So when it comes, it tells you that sample is positive. You don't say it's positive for dengue, you say it's positive for flavivirus. And then you can undertake another step to know which flavivirus it is. You can also be specific and say, I want the primers to detect dengue only. So when you're selecting the primer, you filter and make sure the segment of the virus you're targeting is specific to dengue. And sometimes you can even modify and say it is not just going to be dengue, it's going to tell us whether it is dengue type 1, type 2, type 3, and type 4. But in this instance, we end up using 4 primers. So you have maybe one forward primer, and then you have a reverse primer that is different for each of these types. And you can mix them together and get results. And there are many ways after this to know whether this is dengue 1 or dengue 2. It is some of the things we're going to discuss in this slide. So here is an example of a primer. You can see the length is not so long. Usually it's between 18 to 25 bases. We call them oligonucleotides. So they are fairly short. And remember, the length of the primer and even the content, especially the content of C and G, will determine whether it melts quickly at the temperature that it's supposed to separate, and also whether it attaches at a certain temperature. So the properties of the primer are determined by the content and the length. So different base sequences might have slightly different melting temperatures. With the selection, you need to know the target so that you know how to interpret the results. And right now, when you look at COVID-19 results, you'll find some of them telling you that the primer, there's one primer for subecoviruses. Subeco meaning this is the genus that contains COVID-19 and another virus. Or it can tell you this will be able to detect any coronavirus. So it can detect even the common cold, the viruses that cause common cold that are in the same family as SARS-CoV. And then there is one gene that will be specific to SARS-CoV-2. So that allows you to interpret your results properly. You may find that maybe the group virus, the group part, the group genome, the group gene is detected, but the specific SARS-CoV-2 gene is not detected. That might tell you that this is a different coronavirus. So keep that in mind as you interpret PCRs. You need to know what was the PCR targeting. And the target is determined by the primer. Primer selection is also can be done for different, for the same target can be done differently, and you can have a single or multiplex. For example, you'll find a PCR that can detect 20 pathogens at once, one PCR. So that is a multiplex PCR. You can also find PCRs, which is very common, that can only target one particular genome or gene, and that will be single target. You also, and this usually works, you just find a primer like a forward primer here and a reverse primer that will go this way. And then the target is the area between these purple colors. And you can also have a nested primer to complement this. So maybe you want to be more specific, or you want through the nested primer to identify something else. So after amplifying this in the first round, the first, say, 40 cycles, then you can put in another PCR, but now instead of getting the template from your sample, you get the template from the amplicon, from the PCR that you have run. You get the amplified product, and then run it again in another PCR, and now the next PCR will also have 2 primers. One from this side is the forward, and then another one reverse. So this primer will be called nested because it's within the sequence generated from the first primer. You may hear, as you read nested primers, this is what they mean. So you have run this PCR basically one PCR completely, which is maybe 35 to 40 cycles or even 30. And once you have interpreted your results or you have not interpreted them, you just have the product, you can get that product and run a second PCR with nested primers like these ones. So this allows you sometimes to increase the specificity and also to detect, because now this next primer will have a lot of templates. Remember, you have 68 billion copies. So it is able to detect something that may not have been plentiful in the initial stages of the first primer, of the first PCR. And some people even add probes in between. And this can be added to usually the first PCR run, if you're just running once, to see, to add specificity for certain parts of the genome. So you want to confirm that, say this gene has a certain part that you want the probe to bind to, and then the probe binds, it releases a light, which the machine can detect and tell you that it was present or absent. And we can use that to test for certain diseases. So when you hear of probes, think of that. As I said before, you can have specific or consensus primers, and specific primers are like, for example, dengue only, but consensus primers can be the entire family of dengue viruses, yellow fever, and Zika, which is a family. And sometimes what that means is you may find that the primers you're using are not identical. They have some maybe changes, variations, so that they can look alike. They can be able to, they're not identical, but they look alike, and they're able to identify viruses in the same family. So if you see consensus primers or degenerate primers, think of such primers. But by and large, we are today going to focus on discussing single target PCR, which is what is the primary PCR that we are interested in. The rest I've just put them here for your literacy, for the sake of literacy. Okay, I'll pause here for questions as I can see some of the chat. Could you kindly repeat how the, okay. How does the polymerase know where to stop? Now, the polymerase does not know where to stop. In the body, there are stop codons. But in the PCR we are running, it will run all the way to the end. But usually after the first few cycles, because you are targeting a specific section, the primers will have bound to both sides. And because it is starting where the primer is bound, it will reach the end, which the other primer had already bound before, and so is not extended. It's not extending. So after the first 2 or 3 cycles, they are longer sections, but remember they are limited by the time you are doing the extension. The extension is usually about half a minute. So it will stop, but it will have gone past where the primer will come to attach. So when the second primer attaches, it will shorten that genetic segment to what you are targeting. And so you'll end up with a flash or a double-stranded DNA, which is only what you need. And from then on, it will be amplifying that short section. But initially, in the first few steps, it will have longer segments. I didn't want to focus too much on that because some people get confused with it. Now, stop codons are only in our body, not in this PCR. We don't put stop codons in the PCR generally. Could you kindly repeat how the reverse primer and forward primers work? How will both run from 5' to 3' if the DNA are complementary? Okay, so remember, let me just go back to that first slide, or rather, not first, but. So you see, this is, look at the one down here. These two are complementary strands. The primer for this part is GCCTA. So it will attach from, let me just label it so that you get the 3', 5' thing. Give me a moment, please. So this strand is running from 3' to 5', and the other one is the opposite. So you can see the upper strand is running from 5' to 3', and the upper strand is running from 5' to 3'. The lower strand is running from, on this side, of course, 3' to 5', but essentially it's running this way. Let me make sure my pointer can be seen. So this strand here, now assume this is both DNA, double-stranded DNA. This strand on top here, the primer will not attach from 5' side. It will attach on the 3' side, so that it can be running like this one from the, just a minute, so that I label both. So you can see the strand up here, the primer attached from the 3' side, but that means the primer is then running from 5' to 3' direction. It's just the way the DNA binds. So the complementary strands run in opposite direction. So the primer for this part, let's say the 5' to 3', the strand will begin from here. So it will be A, C, A, G all the way. And then it will attach, assuming the one down here is not there, it will attach, and then the extension will go from now 5' of the primer. So the primer will be identical to this one, this bit. So let's say the first, I don't think you can see my pointer. So the primer will be identical for the first strand, assuming you are making a primer for the top strand, it will be identical to the strand at the bottom. This from, it will now be TG all the way, say, to C, because this is complementary to the top part. And so when the primer attaches, then the polymerase can extend it in this direction, running from 5' to 3'. And this is what we are seeing here, that you have a strand that is running from 3' to 5'. So the primer attaches here, and it is running from 5' to 3', and it goes all the way. And you can see they are not identical. These two are complementary. The two strands are complementary, but one primer is CGGAT, the other primer is TGTC, because they are targeting different parts of the genome. I hope that is clear now. I will continue the questions before I move on to sample collection. The last question was on this 3' and 5', which we've answered. Does one primer move from 5' to 3 and the next one? Well, technically, yes, but you've seen how it works, that you have both primers actually working from 5' to 3', because that's how the polymerase reads. The polymerase reads it. And you've seen how the strands now are read 5' to 3', but in opposite directions. At what temperature do you heat? We'll talk about that, implying that we use a non-primer in a non-sample using PCR procedures. So you know your primer, and you want to know whether your sample has, say, HIV. You don't know if the sample has HIV or not. If it has HIV, the primers will detect it. If there's no HIV, the primers will not attach. Does it mean that you should start with 68 billion in order to get an equal amount of copies? No, the 68 billion is there. It was just one of, if you finish at 35 cycles, you'll end up with 68 billion copies. And you're not even going to count these copies. This is just to show you, you started with one, but how many have you ended up with? Just to show you how much amplification is happening. So you don't even need to remember this number of 68 billion. You just need to say billions of copies. No unknown, not unknown suspected, I don't understand that. I think the primers are recycled and then they're also added in excess. Yes, that's true. Every cycle, you add enough primers, and usually in optimization, you know what you need and the quantities, there are calculators for this, to know that I want this, for this primer, I want this concentration. And so you put it at the right time. But then that's why we don't run PCR for 100 cycles, because then you will use up all your resources, the DNTPs, your enzyme, your primers. But if you're running 40 cycles, we keep reusing these primers because they attach and they anneal. So when we melt at 96 degrees Celsius, even the primers will detach, they will be denatured. Well, not when we anneal, when we denature at 96 degrees Celsius, the primers will detach. When we bring it back down, maybe to 60 degrees Celsius, the primers will attach again. And now they're attaching to whatever template is available. And then we take the temperature a little higher to 70, and then at that temperature, the polymerase extends, copies that segment or that strand that has attached to a primer. And then we denature again. By denaturing, the copy, the double strand that has been made or copied by the polymerase will separate. The primers will also separate. And then you can repeat the process over and over. I'm reading your questions again. I don't think you can recycle the primers if they already have been added to the suspect. You recycle them in each cycle, not after you can't take them to the next test. But for each cycle, for each PCR, you keep using the primers that are in there. But remember, if they are not, if they do not get extended, if they do not, they were not. This is happening in like a bowl of soup, and there are so many primers in there. Some will work, will be used, some will not be used. So those that are used and incorporated will become part of the genome, yes. But those that are not used up by the polymerase to extend will now become in the next cycle, they may be used or they may be spared. But after 40 cycles, you've noticed that most of the primers have already been used up. They cannot be used again. They cannot be, there's nothing to anneal. And if the enzyme has been recycled many times, it gets tired, basically. It gets exhausted and cannot work very well. And the DNTPs can also run out. So you don't have anything. So after 30 to 40 cycles, you have exhausted all your resources. But remember, the primers are in the mix at measured concentrations. So for the first 30 to 35 cycles, you have enough to use. And every time they attach, if there is no polymerase to extend them, they will anneal. And then they can, they'll be denatured during heating, and then they can anneal for the next cycle. And then, but once they are used up, then for the next cycle, they'll not be available. Okay, I feel like we need models to show how this works. How will you extract? Okay, what is a probe? I will let you look at that. It's not very important. I have not understood how nested primers and probes. Could you please recap? Nested is basically inside. So you are targeting a portion in another PCR within the initial portion that you are targeting. So if you are targeting from position 1000 to position 2000, your nested PCR can start from position 1200 to 1800. So that is a nested PCR. What do you mean saying not the primers aren't incorporated into DNA? Yes, they are, but not all of them, because they are sufficient. But they will still attach. They will still anneal. But if they are not extended, they are not part of the new copies, they may not be. If they are not extended, they will not be part of the new copies, because there is a lot of primer, and sometimes they even bind to each other. It is called primer diming or primer dimers. So the primers are there, they will bind and anneal until they are used up. They'll keep binding and releasing until they are used up. And sometimes when they don't, when they bind to each other, they release, and next time they can bind to something else. Okay, I think somebody has asked and clarified. I think I'll continue with the lecture now. We'll come back to the questions. Now, specimen collection is as important, and that's why I'm insisting on discussing it. If you're collecting an RNA virus like dengue, you have to be careful that even SARS-CoV-2, that your samples are not exposed to heat for too long, because the sample RNA viruses are very fickle. They'll get destroyed because we have a lot of RNases. RNases are enzymes that break down RNA. Even in your fingers, you have RNases. They're everywhere. So RNases will be everywhere. And if you don't freeze your samples quick enough, they may destroy all the genome, the RNA virus, and then you will not be able to detect it. So yes, we can test whole blood, we can test serum, we can test plasma. My favorite is the EDCTA tube, because it gives you plasma or sometimes whole blood if you don't centrifuge. And that is, you can use it for testing, and you can also use serum. But also, for example, RNA viruses, there are now commercial RNA preservation tubes. You may hear of things like Tempus tubes or Paxgen tubes. You don't need to remember the brand names. The idea here is that the RNA preservation tubes, which are filled with some buffers and RNase inhibitors. So when you put the sample there, it does not break down. So this helps you to collect a better quality sample. So that tells you that sample collection is important and you need to pay attention to it. If you're in the lab, I mean, in the casualty department or somewhere, and you want to test for HIV or for polio or for measles, if you collect a sample and it's not kept well, or it stays in the bench overnight and goes to the lab tomorrow at 10, then it means you're not going to get the right results. So keep that in mind. It's just like collecting what? A urethral swab or a high vaginal swab, and you want to test for gonorrhea. If you put that swab on the table and then it's going to be tested tomorrow, then you'll not get anything and you'll say the patient does not have gonorrhea. But if you pick that swab and put it in a transport medium or apply it directly on a agar plate and then take that plate into an incubator, then you're likely to get the correct results. So think of the same as bacteria. I mean, viruses that they cannot survive, especially the virus. The RNA viruses cannot survive for too long, so they need to be taken care of. The DNA viruses are a bit different because DNA is very hard to destroy. So hepatitis B, HPV, herpes, those are likely to be there. The DNA is likely to be there because DNA is not so easy to destroy, but RNA is different. Okay, unless you're using dry blood spot, because in dry blood spots, the RNA will be captured there and there's no fluid medium in which RNases can act. So you still be able to retrieve some of the RNA. So one person asked a question, how do you get DNA from RNA? So you start with your isolation. Isolation is getting nucleic acids from your sample. So you're starting with blood. You want to get the nucleic acids from that blood. And you're extracting all the nucleic acids, the host, if you are targeting HIV or dengue or whatever other pathogen is in there, you will extract all the nucleic acids. At this point, there's no selecting, and there are processes. This is one of them. It's column-based. This column, you put the sample here, and with several reactions, with some solvents, you spin it, and then it will retain the nucleic acids in this silicone filter, and then eventually you will elute it away by using alcohol. To basically wash off the DNA of the nucleic acids, could be DNA or RNA, into a container. And usually you get from, say, a sample of 500 microliters or half a milliliter, you can end up with 50 microliters, which is a very small volume, but it's enough because a reaction can take even 10 or 20 microliters. So this is column-based. There are other extraction methods. Some are even bead-based. You can extract using magnetic beads. But the good thing is that each kit will come with instructions. But the idea here is whatever method you use, you follow the instructions and you'll end up with nucleic acids. If it was an RNA virus, you know that the nucleic acid will be RNA. And if it was a DNA virus, then the nucleic acid for that virus will be DNA. So you can do additional steps, although not everybody does this. I also don't always do them, like purifying the nucleic acids by removing buffers and other solvents, or quantifying to know how much you have extracted, and there are things to do for that. So those are optional. When you read and you see them, don't worry too much about them. So after you get your nucleic acid, you can then make cDNA, which is complementary DNA, through reverse transcription. And this is basically just putting the nucleic acids in a tube, and in that tube you add reverse transcriptase, you add RNase inhibitor, you add DNTPs and primers. Usually the primers are general. They're not specific to your virus. They're general to, say, we call them random hexamers. They can bind anything, so they'll convert most of the nucleic acids into cDNA. But if you are you want to test herpes or hepatitis B, because you are starting with DNA, you don't need to do reverse transcription. So reverse transcription is only for RNA viruses like HIV, polio, measles, dengue, and what have you. So if the virus is already in DNA form, the genome is in DNA form, you don't need to do this step. And also now there are some PCRs we call one-step PCR kits that come already loaded with the PCR reagents, but also the reverse transcription reagents, which means they will, in one step, you can do both reverse transcription at a different temperature. Then you inactivate the enzyme using temperature again by raising the temperature to maybe 95. The reverse transcriptase is killed or destroyed, and then now you can proceed to PCR. That means the person making the kit always considers which reverse transcriptase to use and what temperature it will be denatured. So the product you want is DNA. We call it complementary DNA. And that is what you take to your PCR. So remember, this is done for RNA viruses. The actual PCR will be like this, like we discussed before. This is just for repetition so that you understand. There is the initial step of reverse transcription. You can see we are doing it at 50 degrees Celsius, which tells you that the enzyme reverse transcriptase that was selected in this kit works at 50 degrees Celsius, and you're only doing one cycle for 30 minutes. Then you denature. By denaturing here, you are heating at 95 degrees Celsius for 15 minutes. This denaturation will destroy the initial reverse transcriptase enzyme so that it doesn't continue its work. And it will also denature the DNA from double-stranded to single-stranded. And then now you go into shorter cycles of 1 minute each, where you have, and this can change, these times can change, they don't have to be 1 minute. Even the temperatures can change slightly. I'll tell you why. So the denaturation will now occur again for 1 minute. Then you allow the primers to bind. So the primers will bind. Remember, you are starting with low templates. So some of the primers will not bind initially, some will bind, some will even bind to themselves. But every time you hit, any unused primers will get denatured. And then they bind again when you cool down. You can see we have cooled down from 94 to 54 degrees Celsius, for example. It could be 50, it could be 60. And after cooling down, the primers attach or anneal. And then after they attach or anneal, the polymerase now attaches to the primer. And when you take the temperature to 75 degrees Celsius, it extends by that, it copies that strand into a double-stranded genome or the double-stranded DNA. And you can see here the end product is double-stranded DNA. In the next cycle, remember that was one cycle. In the next cycle, you hit again, they separate, the primers attach, the polymerase extends, you end up with another set of double-stranded DNA. And this is repeated 30 to 40 times. And eventually, you finish with a final extension, basically of the polymerase doing its work for about 10 minutes. And then after that, if it is an endpoint PCR, you can visualize on a gel like this one. But if it is a real-time PCR, I'll show you how we visualize. And what we do with gels, I think maybe I should show you the gel, that's how it looks like in the video, you saw it, but I'm going to do that again. Just a short brief bit on the video. We saw we saw in this video, I hope you can see the screen, if you can't let me know.
PCR finishes depends on our experiment. We may insert it into a plasmid so that a bacteria will produce a certain protein that we are interested in. We may use it to do DNA sequencing so we can learn the exact sequence of nucleotides in our DNA sample.
So this here is your gel. And you can see the gel is basically some powder. You prepare it in a microwave by mixing with water. You cool it down in this trough. This is a trough. And it allows you then to, if you put a comb, it makes wells. You can see these yellowish or orangish holes on the gel are called wells, where you put in your sample that already has a dye, and the dye is usually binds to DNA. And when you run electricity from this side to this side, the gel, the bands will flow. Basically, the DNA will flow. And when you take a photo of this, or when you visualize this with a UV imaging device, you can see the bands of DNA. If there is no band, it means that sample is negative. If there is a band, that sample is positive. It could be a positive control. It could be the positive sample itself. So I don't think there was much.
Often, we will run it on an electrophoresis gel so that we can visualize the size.
Yes, so that, I just need you to see that, that gel, how it looks like, so that when talking about gel, you're not too lost. But there's a video here to show you the full process of making a gel if you're interested. So you see here, these are the wells, and these wells are made using a comb, because the gel is usually liquid. It's like very liquid initially, but as it cools down, it hardens. Not as hard as rock, but as hard as a what? Those of you who have eaten halwa or gelatin. Yes. So that is these wells, you put your sample. The first one is a ladder. The last one is a ladder. This one is empty for this from this mini. So these are ladders, and the ladder will tell you the length of your band. So the smaller bands will go the farthest. The longest bands will be heavy, so they'll travel a very short distance. They all start here, and then when the current is passed, the bands will migrate over a period of 30 minutes to 1 hour. And then because you have the dye, you have seen a dye that is there, you will see them migrating so that they don't, if you keep running, they can run out of the gel. So you make sure you see when they are giving you enough distance to differentiate the different bands, then you can stop the electricity and take this gel to a UV light imager. Then you look at it and see the bands, the different bands at the different lengths. And here you can see your ladder, and this is a positive control. In this case, this is a gel I took. I was testing for, I think, alpha viruses, and we used a ladder. Alpha viruses are, I don't know if you guys can see my pointer. Alpha viruses are things like chikungunya. And you can see what we used here has different bands, but the one we're interested in is this one. And this is around 200 base pairs because the target segment, the target DNA segment was 200 base pairs long. So one primer was starting, say, at 1,000 base pairs, and the last primer was stopping at 1,200 BP position. So if you see a positive like this one, it will be corresponding to that one. If there's a positive and it is not, maybe it is 1,000 base pairs, then you know it is not positive. It is a false positive because it amplified something else, not what you are looking for. But what we did after this was to sequence using something we call Sanger sequencing that we'll discuss next time to confirm what this is. Because remember, if you're using group consensus primers, like you want to target any coronavirus, then after you get a positive, how do you know which coronavirus? You go the next step and then sequence this with Sanger to tell you what the sequence belongs to. And so this is a positive sample. The rest are negative. What you see here are what we call primer dimers. These are when the primer attached to each other. They also produce bands, and you see them at the end here. So these are not positive. You can see negative control here, for example, as a band. So if you don't see these ones, then you know maybe you did not put a sample in your well. So these are like to show you where your sample has reached on the gel. So all this is the lightest. You can see it's only 50 base pairs, which is very small. So each of these well is an individual sample. All these samples are negative. You can label them if you like, but I only label this one with very few so that you can see the positive one, the negative control, and the positive control. This negative control could just be water, but you see it still gives you some bands of what we call primer dimers, which is basically the dimers, the primers joining together, a non-specific binding, which then produces this kind of thing. Okay, so this was what I was just telling you, Sanger sequencing, where you get allows you to get this sequence. And if you copy this sequence and blast it, you go to this database, it will tell you that sequence belongs to a human being, that sequence belongs to a cow, that sequence belongs to a plant. And that is a topic we'll discuss at some point. So I don't know if I've skipped something. Let me see. I think we're okay. The other thing I would like to highlight is real-time PCR. So sometimes it's called RT-PCR, but some people will use RT to mean reverse transcriptase PCR. So be careful. And real-time PCR is also called qPCR or quantitative PCR, although it can be quantitative or semi-quantitative. I'll show you what that means. So based on the same PCR as conventional PCR or as we've seen. But what I have shown you before is called endpoint PCR. You see the results at the end. For real-time PCR, you can see the results as they happen. If somebody has a very high viral load, you can see in the curves coming up, you can see a positive test very early, like the positive control can come up very early. And if you're running a PCR, you can even call and say, oh, this guy has COVID or has Ebola, for example, if you're using real-time PCR. And because it monitors the amount of the target DNA after every cycle. So remember, you're running 40 cycles. So after cycle 1, the reagents are mixed with a fluorescent dye, so that every time the double stranded DNA intercalates or comes together, a signal is released. And that signal is detected by the machine, and it is drawn into a curve, depending on the amplitude of the signal. And that is the curve you can use to say, depending on how quickly the curve has come up, to say if the sample is positive or not, depending on when the curve crosses the threshold. I'll show you an example of a curve. And maybe I should do that and then come back to this. So these are the curves I'm talking about. So when you start, and this is really a simple chart, on the side, on the y-axis, on the vertical axis, you have fluorescence. Remember, this fluorescence is being released by a dye which has been put in the reagents, so that every time DNA intercalates, the dye is released. You see the signal. The computer detects the signal. On the y-axis, on the vertical, the horizontal axis is the cycles. You can see this goes up to 35 cycles, from 0 to 35 cycles. At the first cycle, you can see there is no detectable fluorescence, but you can see some beginning to be detected at around 8 cycles. And then the horizontal line you can see is called the cycle threshold. And this is usually calibrated on the machine, so that when this curve crosses this threshold, then you say that is what you call the CT value. And if the CT value is of meets a certain criteria, then you can say that sample is positive or negative. But you also use the CT values for quantitative purposes. So for example, this one that has come up very early means that the DNA is a lot. So this is, if you can say, very positive. It has a higher viral load than B. B has a higher viral load than C, and C has a higher viral load than D. So what about E? E has come up at around 32 cycles. Is it positive? Is it negative? So when you optimize your PCR test, you'll have a negative control, and you'll see even the negative control come up at some point. So after so many cycles, even the negatives will come up because of the primer dimers. So like this one would say is negative because maybe the cutoff for this was 30. Maybe we had optimized the assay and said the cutoff for this PCR is 30. For some PCRs, the cutoff is 35. For others, it is 40. So for this one, this is negative because it has come up too late. But the others, this is 23. This is the cycle threshold. You read here 22, sorry, not 23. You read here, this is about 18. This one is about 15. This one is about 10 or 11. So the idea is you have quantified by CT value, but if you have standards, if this was standards and you know this standard contains 100,000 copies, this one contains 10,000 copies, this one contains 5,000 copies, and D contains 1,000. If a sample comes in between here, you can use a computer model to calculate the viral load exactly. And you can say this has 10,000 copies, for example. So that is quantitative, but the CT value can be semi-quantitative. So if somebody had a COVID test and tells you my CT value was 10, then it means that person has a very high viral load. If they tell you the CT value was 28, then you know the viral load, depending on how many cycles were run, the viral load was present, the virus was present, but the viral load was not so high. So that is what we mean by quantitation. And this is what we meant by the data being plotted on the screen in real time. And you can see here that the idea is to get your viral load or the CT value, which is a threshold after which you can say the sample is positive or it has reached that threshold. And this does not wait until your PCR is over. You can see at every cycle, it will visualize for you. So this will begin to come up at 8 cycles. It is not positive yet, but the moment you see it coming up so early, you know it's likely to be positive. And you can even, once it crosses the threshold, you can even call and say this person has that disease that you're testing for. I hope that makes sense. So this is called a real-time PCR, and these are called melting curve. And what we've just done is melting curve analysis. And sometimes, depending on your on what you want to test, you can use this kind of PCRs to test different things. So when you're talking about the multiplex assays, most of them are real-time PCR. What could go wrong? Pre-analytical, meaning before the test. Sample collection, as we explained earlier, can be an issue. The storage, how you store your sample after collecting. For example, I was doing research in Kibra and in Taita. The samples in Taita, we had to collect them the same day and store them at a freezer, which was -20. If you leave it out there, it will not be a good sample. And then there's the headache of getting it from Taita to Nairobi. Remember, there's sun, we are going through Kambani and Voi and all that. So you have to find a way to transport these samples to Nairobi without exposing them to a lot of environmental degradation. So we use dry ice, which is takes even 24 hours or more to melt, and so that we got the samples here on time. Primer selection can also be an issue, but that is something the lab has to deal with, but you keep it in mind. Unless you want to get into research, you may not necessarily need to select a primer yourself. Analytical errors could be pipetting errors. Now the volumes we get, for example, the volume we put in a single tube for DNA polymerase is so tiny that you may not even see it, that you may not even know that you have dropped it into the well. So pipetting errors can occur. You see, if you don't put the enzyme, nothing will happen. Contamination can also occur. Now, especially if you tested a positive sample, remember it is 68 billion copies, and then you release that into the air by opening the tube. It can even contaminate your kits and other samples. So what we do is separate the procedures so that in the place where we are doing the amplification, we do not do any other process except maybe gel. The other processes are done in a clean room where there's less chance of contamination. The post-analytical errors could be interpretation. Remember, for example, this one. Somebody might read E and say this person is positive. Yet we have agreed that depending on the optimization of the kit, you can decide that everybody with a CT value of more than 30 will be considered negative. But the person in the lab has to make that decision every time. So what if somebody comes and reads this, oh, this one has also come up. It has come up late, but it has come up. So they will say that person is positive, yet it is negative. So post-translational post-analytical errors can occur. And transcribing. Sometimes you are reading this on the chart and you want to type, and you are typing there to say, you can see it is negative, but when you're typing, you type positive. Can happen. So that you need to keep that in mind. Although there's something we call quality control in the lab, I think you're going to talk about. Somebody usually counter checks to make sure that what you have written is correct. Quality control, yeah. Workflow and design, I'll show you a good workflow for PCR. Competence, training, and experience. You can't just do this test unless you're trained and experienced. Non-sample as control. So we usually get non-samples as controls. So that if and also we get water. So if your water comes up as positive, then you know you blundered. If your positive sample comes up as negative, then unajua kuna shida. Internal controls for some kits. And if you guys have seen Gene Experts, they are very good internal controls for their kits. Also, in fact, for Gene Expert, if you put the wrong sample, like if you're supposed to put sputum and you put urine, it will not read. It has those controls. External quality assurance programs are also established, but this will be done as a topic later. So the idea here is, for example, at the Kavi Institute of Clinical Research, you have a research lab, and we are enrolled into different external quality assurance programs, where every 3 months or every month they will ship samples that we don't know the results. And once we test them, we send the results back, then they tell us out of 10, you got 10 or you got 2 or you got 3. Most of the time we get all of them, but once in a while you may get a problem, especially during COVID. There were issues with shipments, or the samples were in bad quality, so we got a lot of false negatives. And so that that allows you to know whether you're doing the right thing or not. Now you don't have, because the external quality assurance programs may be expensive. You can also quality control using interlab comparison. So for example, we have a lab at Unity, at the college or at the faculty, and a lab at Kavi. Kavi can decide, we'll be testing 100 samples of TB every day. Out of those, we'll send you 10. We will not tell you the results. We'll send the other lab 10, then they test, and then we compare our results. So that allows you to tell whether you're doing a good job or not. And then you can also do lab repeats. You already have your samples that you kept. Every time you're running or you get a new kit, you test those samples and see whether you get the same results. The problem here is that you have to make sure the storage is good. Otherwise, you end up with a sample that is degraded and you think it is positive, kumbe it has, the DNA or the RNA has been degraded. This here is a lab layout, and you can see here you have a separate room for sample storage, you have a separate room for extraction, you have a separate room for PCR setup, and this room is very important, where you put your enzymes and your other reagents. And then you cross over to the next place for PCR amplification. This is where the machine called the thermocycler, which cools or heats to 95 degrees, very quickly cools to 50, very quickly warms up to 60 or 72. That is called, that machine is called a thermocycler. And the post-PCR work could be things like gel electrophoresis. Sometimes you can also purify and do other things. And then you need your office space and other areas. So this layout is important because if, for example, you get something from the amplification room, like a pipette, and take it to the PCR setup, you have already contaminated your kit. And that means that kit will always give you positive results, and you have now to throw it away and get a new kit, which is money lost. Or if you take it to the sample storage, it means you'll contaminate all the samples and they'll all be testing positive. So because at some point you have to aliquot samples using the same pipettes. So generally when something leaves these rooms to this side, it doesn't go back. But you can pick anything from here and take it to this side. That's it. I have a few questions, then I'll answer yours. So tell me in the chat whether this is true or false. I can see somebody has already sent true. I'm just kidding. So the first question is, CT levels are inversely proportional to the amount of target nucleic acid in the sample. I can replace that with the viral load. So our CT value is inversely proportional to the viral load. I can see true, true, true. There's no false. Okay, you guys are correct. That is true. The next question is, the tag polymerase is denatured at 94 degrees Celsius. This is very interesting. So it's number 2. You can write there 2, ndio nijue unajibu gani. 2 false or 2 true. 2 false or 2 true. So I can see some false, I can see some false. And you guys are correct again. The tag polymerase is able to withstand high temperatures, so it will not be denatured at 94 degrees Celsius. What gets denatured at 94 degrees Celsius is the double-stranded DNA that separates, basically. Okay. The third question, a primer determines which DNA segment will be amplified. A primer determines which DNA segment will be amplified. Is that true or false? I see a lot of answers, true, 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 true. And that is true, yes. So I think you've understood that. I will now look at the chat and answer or respond to your questions. If you have additional questions, you can still post them in the chat. Okay, thank you. Those answers are enough for now. So, oh, I see. How will you extract a template DNA if there is none? True. When you're extracting a sample. Say you take a blood sample. You're extracting all the nucleic acids in that blood sample. You don't know whether the HIV you are targeting or the dengue you are targeting is there. So you extract everything. In fact, that sample you have extracted, if you wanted to test for human genome, you can test. If you wanted to test for RBC, whatever, genes, you can test. But now in that case, you want to test for HIV. So you can't tell. Only the primers will eventually tell you you have to go through the entire process to know whether the virus you're targeting is there. So you mean to say the primers are not incorporated in the new DNA? I think we had answered that. Oh, yeah. The recycling is not one for one test. How are primers themselves duplicated since we are so many for PCR? Primers are not duplicated during the PCR, we make them before. Like you have seen things we call DNA synthesizers that are they make these oligonucleotides or these primers. So when you're putting, you put enough. When you're adding to the reaction, you calculate, there are calculators, you know, for this one I require this number of millimoles, this is the volume. If you guys remember chemistry, there was a formula we used to say C1 V1 times C2 V2. So you have to do a bit of maths, but that's okay. Deoxynucleotide triphosphates, what is DNTP? Yeah, DNTP are basically the ACTG for DNA. And there is one area where RNA has 2 hydroxyl groups, DNA has one of the hydroxyl groups removed, it has an oxygen without hydroxyl. So that's why it's called I mean, it has not an oxygen, it has hydroxyl group removed, and that's why we call it deoxy. And ribonucleotide is not deoxy, it's just ribonucleotide. So this is deoxyribonucleotide. It's a word that sounds like a tongue twister. I thought it had something to do with timing. So all of them, S, C, T, and G, can be deoxy. And in fact, next time we'll teach you about dideoxy, which has removed both. Okay, speaking of recycling, does it mean that once you extend the first DNA strand and separate them, the primers break away? No, they don't. They've already been incorporated, so they will now be part of that. But the ones that are not bound, the ones that are not bound to the ones that have not been extended, they will not detach. But there are some that usually bind to each other. That is called primer binding or primer dimers. Those can separate and attach to other things, although sometimes at the end there'll still be some of them remaining. Hapo kwa gel nimepotea. Oh, okay. Gel is just something for visualizing the DNA. And what is happening is that you are using the gel as a medium to allow you to separate the different sizes of DNA. Because you have now plenty of DNA, you put each sample in its own well, and then you pass the current, and these samples are now the ones that you have amplified with PCR. And when you pass a current, they'll migrate to the one side of the gel, and during that migration, different sizes of the DNA will move at different speeds. And for you, you are looking for a specific size. Like an example I gave you, the PCR that I showed you, we were targeting a band of 200 BP or base pairs. So we looked at the 200 base pair using the ladder, and we can see at that point whether there's a band or not. If there's no band there, then you know the sample is negative. Why do we use serum or plasma or to extract DNA? It's not serum or plasma. You can, because if the virus is in free form, you can get your virus in plasma. But for example, if you are doing influenza and you take your blood, you will not find any RNA for influenza there. You take your nasal swab or nasopharyngeal swab and you use that. If you are testing for herpes virus, you take the fluid in the blister. So the sample will be determined by what you're targeting. Since the CT value is a straight horizontal line, how are we using it to quantify? Very good question. I'm going to show that again. I hope you can see my screen. So I'm going to have to share this again. Okay, so I'm now just sharing my presentation. And this here is the band. The threshold here is a cutoff line. So that if any sample goes above this threshold, you can see this is one. It is considered to have detectable double-stranded DNA. Detectable double-stranded DNA. And the earlier the DNA is detected, it means the more the template you had available. So the CT value is not this value here. This is the cycle threshold. It is the line that determines the CT value. So if you come for A, the CT value is where the line A crosses this threshold. And this is around 10, or 11. For B, it crosses the line at 15. So the CT value for B is 15. And those are just cycles. It crosses the value at the 15th cycle. The C crosses this line at the 18th cycle. D crosses the threshold at the 22nd cycle. And remember, the earlier it crosses, the more the viral load. So if something crosses at the 10th cycle, it has a CT value of 10. It has a higher viral load than this one that is crossing at 22. I hope that is clear. Another question is, what advice do you give to someone interested in lab diagnosis? What is the career path? Kamunya, there are many career paths. You can become a microbiologist, you can become a pathologist, you can become, different things. So the career paths are different, depends on what you want to do. Even radiology has elements of diagnosis, although it's not necessarily lab diagnosis. In layman, ile iko mingi itaonekana haraka. Yeah, that is true, Nanga. Ile iko mingi itatokea haraka. CT in full is cycle threshold. Okay. What is question? Why is question 1, 2? I thought it's directly proportional. Okay. So the CT value, the lower the CT value, the higher the viral load. Okay. So I think we should be okay now. I see no more questions. This is PCR. It's better, of course, when you do it, but this allows you to understand it better as you read, and also to think through when you're reading patient results and look for what genes were they targeting. It's not just SARS-CoV-2. You might see that they were targeting this gene or the other gene. How do you interpret those results? Okay. So how come the double-stranded denatures at 94, yet the denaturation step occurs at 94? Yes, so the reason we are taking this 94 is so that we can denature double-stranded DNA. Why? Because the primer will only attach to a single strand, and the polymerase will only extend a single strand. It does not happen when the RNA, the DNA has not denatured. So when the DNA denatures, it denatures at 94. So denaturation occurs, and that means you have split. And then you lower it to a temperature which allows the primer to, which is around 60 degrees, in this case 54, to attach. And when it attaches, it primes the polymerase to work. But you know, the polymerase will start working at the optimum temperature at 72, which then allows you to have double-stranded genomes. So at 72, your double-stranded genomes will still now be intact, the ones that have been copied. So for you to separate them and use them for the next cycle, you have to denature again at 94. So you keep repeating this cycle. You denature at 94, you attach your primers, you extend, you go back to 94 again. But this is not done manually. You set a machine that keeps doing this in an automated fashion. Once you set the machine and you put the samples, you just come back after you have the PCR has ended, or if it's a real-time PCR, you can look at the screen to see what is happening. I hope that is clear now. Can you repeat the gel electrophoresis mechanism? Sure, we can. I see, I think I don't read part of the question. How do we identify presence of viral DNA? Okay. So what happens with the gel electrophoresis is that you have the sample from the PCR, the amplicon, you call it the amplicon, and you mix it with two things. One is a dye that we use to visualize. And that dye you saw is either green or yellow or whatever. Excuse me. And you also mix it with, so the dye you can see, and you also mix it with something that binds DNA. There are different names. One is called gel red. There's another one called a bromide compound, which sometimes people consider able to cause cancer. It's called ethidium bromide. And the idea is you're mixing those two things. The first one, the dye, is for you to see. You can see with your eyes when the samples are flowing from the wells all the way to the other end. And if you don't stop it, they can actually flow out of the gel. So you see and you stop. When you stop, you cannot see the DNA still. The DNA is bound. It has been flowing, but it's bound to, say, the ethidium bromide, the dye that binds to DNA. So for you to see it, you have to use an ultraviolet imager. There's a gadget, we call it UV. It uses UV to shine on this gel, and then you see on a screen all this. This is taken now from the screen using the UV light. So the imaging system is a UV system, and you are able to see the DNA because it is bound to the compound that can release light under UV. And this compound in this case can be ethidium bromide, which some people don't like because it's said to be cancerogenesis or causing cancer. Or you can use gel red, which is thought to be safer, and there are a few other dyes. So I hope that is clear now. If you are not sure, look at this video, and if you still don't understand, you can ask more questions next time. Okay. Yes, carcinogenic. Thank you. Carcinogenic is what I was looking for. Mohammed Abdirashid, you have saved the day. Brain ni kama ilikuwa imeenda holiday. It is derived from thermophilic bacteria. Oh, Taq polymerase. Why can't it be natured? Because it comes from, we are taking advantage of nature. It comes from this bacteria that can survive at 94 degrees Celsius. So it has that enzyme that cannot be denatured. Okay, so I think we can stop there. Thank you very much for your attention. I glad we are finished on time. For those who want to go for prayers, you guys enjoy your weekend. We'll see you next time. Santeni.
Thank you.
There's a question on the optimum temperature for Taq polymerase. It's not 54, it is 72.